correcting some misconceptions about chromosomes as previously studied in some courses and curricula, as well as which you studied in undocumented internet pages, which far from specialized textbooks and scientific research. So let's start it together. بسم الله الرحمن الرحيم. هنحاول نعمل تصحيح لبعض المفاهيم اللي للأسف قد درس بعضها في المقررات والمناهج الدراسية أو التي على صفحات النت بشكل غير موثق وبعيدة عن الكتب المتخصصة والأبحاث العلمية. فهنبدأ مع بعض إن شاء الله بعض هذه المفاهيم وتصحيحها. So let's go. Don't be confused. This is a chromosome. This is a chromosome. Okay? One chromosome. That's two chromosomes, or duplicated chromosome, or metaphase chromosome. So that is the one chromosome, and that is a metaphase or duplicated chromosome. Alright. The structure of one chromosome, which found on your left hand, consists of two arms. The short arm called B arm, whilst the long arm called Q arm. However, both shaped of a chromosome before metaphase and the chromosome at metaphase are the following structure. Both have centromere, but in the case of metaphase chromosome, such centromere coated by kinetochore, which is a proteinic substance, and in turn attached to the spindle fibers during the cell division, mitosis, or meiosis. Also, the duplicated chromosome or metaphase chromosomes consists of two sister chromatids. Please don't forget this term. Two sister chromatids, because they are identical in genetic materials. Okay, this is a copy from this. Okay, this a copy from this, and formed at S stage during cell cycle of a cell, and in this stage is found in interphase stages. Okay, at which the DNA replicates into two strands, each strand duplicated into two strands. Okay, so this is a form this shape of chromosome, duplicated or metaphase chromosome. Why it is called metaphase? Because it is the appearance one shape of the chromosomes during cell lifespan of the cell. Why it is duplicated? Because it has 2 copies of DNA, identical genes are found in both 2 sister chromatids. So when a gene called like that T capital, this is the shape or the case of allele for this genetic trait. In another one, in the another sister chromatids, it also T capital letter like that. When it is T small recessive character, also this is, it will be T small letter. Okay, or type. Okay, whilst in this one chromosome, it is a half of 2 homologous chromosomes. Okay, has this is derived from mother gamete and has also another 1 chromosome identical or homologous to it, but from father gamete, at which both chromosomes are called 2 homologous chromosomes. When this form of chromosomes have T capital as a genetic trait, it is not entered in another homologous chromosomes has T capital letter or dominant characters, but it may be another homologous T capital dominant character or T small recessive character in another homologous chromosomes. Okay, also both shapes of the chromosomes has a centromeric region, which is called primary region, primary constriction region. Primary constriction region, also this is another name of centromere. So the secondary constriction regions may be found at this region or this material, forming satellite unit of this region. Okay, after constriction, secondary constriction, while it's this is the centromere is named primary region or primary constriction of chromosomes. Okay, you note that there is 2 colors, bluish coloration. One is a dark, which represents the heterochromatin. Hetero meaning a changed chromatin, whilst U meaning that true chromatin. Okay, this is containing the functional or active genes, euchromatin, whilst the heterochromatin containing non-functional genes, or may be called junk genes, or inactive or non-functional genes. Okay, Both shapes of chromosomes have telomeres. Telomeres are end region at the end in both ends of two shapes. Telomeres. You can return to the lecture of telomeres to identify more informations. Okay? Please don't be confused between the two forms. Classification of chromosomes based on the position of centromere. Now we will deal with the shapes of chromosomes according to the location of centromere. At first one, this shape of chromosomes called metacentric. Metacentric may be in the center of chromosome, the centromere region. So, the centromere region located at this position, middle site of the two arms or two sister chromatids of metaphase chromosome. The second one is called submetacentric chromosomes. Maybe that is two arms, up two arms, smaller or shorter than the Q arm. So the B arm is shorter than Q arm. And the centromere up slowly to direction of P arm. The third case of this classification of chromosomes according to the location of centromere, at which the centromere up more than the submetacentric, and in turn it is called acrocentric chromosome. Okay. In fourth case, this is called telocentric, whereas the centromeric region are found at the tips of two arms. There is no at which B or Q, just taking the this shape, like that chromosomes which are found in mice. This is a good example for this type of chromosome. To conclude that differentiation between the four shapes or types of chromosomes, we will say again, first one is called the metacentric chromosomes, centromere at middle position. Submetacentric, at which the centromere up slowly than this. Third case, acrocentric, the centromere up more than the submetacentric, and finally the fourth shape, which is called telocentric chromosome, at which the two arms are derived from one centromere and there is no P arm. There is also misconception between the following terms: homozygous, heterozygous, homozygous. What is the difference between them? Okay. Now, firstly, what is the meaning of homozygous? homozygous. Homozygous means both alleles present on the homologous chromosomes for a given gene are the same. Both alleles, like that. This is two homologous chromosomes, and that letter and the another letter are called alleles. Okay, these letters represents the alleles. So what is the meaning of alleles? Allele meaning two or more alternative forms of a gene. Again, allele meaning two or more alternative forms of a gene. Okay, this is the two alternative forms of the gene. P, which maybe represents black fur of mice, rabbit. Okay, and that is the two forms of a gene B. Gene B, which meaning like, for example, white fur of say mouse or rabbit. So the allele is B. That is the two forms of such allele, and that is the two forms of such allele. Okay, so the two forms in this case involved BP. Both are capital letters, whilst in this case B small and B small, this is the two forms of allele. Okay, homozygous meaning both alleles for that gene represents the color of fur as a genetic trait, take two forms, B, two recessive characters, whilst the two recessive characteristics in the case of another two homologous chromosomes, such as B capital, or two genes represents a trait of dominant character like black fur, as we mentioned. Okay, that is homozygous chromosomes. Homozygous chromosomes. Both alleles present on the two homologous chromosomes. So, when we study what is the meaning of heterozygous, hetero meaning difference. There is the two alleles of that gene are different from each other. One is a dominant and another is a recessive characteristic. Okay, so the two genes are as you see, one is a capital letter and the other is a small letter. One is which we say dominant characteristic, and the other is a recessive characteristic. Let's go to the third term, hemizygous. Hemi just only meaning that's half. There is a half of both alleles. Say, when you suggest a such example which represents the two chromosomes as in case of karyotype of any male eukaryotic diploid males contain one of chromosomes is X, or that is taller, and the smallest or shorter chromosome is represented by Y chromosome. Okay, so the genes which are found have how much alleles? How many of these alleles? Yeah, of course, just one allele. And these genes represented with just only one allele, because this is represents the masculine characteristics, and this chromosome represents the which is called X-life chromosome. Okay, in case of male organism. In another example, as illustrated in this figure or this diagram, one of two homologous chromosomes has B capital letter, whilst the other is deficient, as illustrated in this figure. So in this case, we name this phenomenon by hemizygous. You can now differentiate between homozygous, heterozygous, and hemizygous characteristics or genes. Now we are going to study the homologous chromosomes and non-homologous chromosomes. Starting to differentiate between them, we must study what is the meaning of homologous. Homologous meaning or describes the relationship between two chromosomes are the same or different. Okay, so homologous chromosomes refer to a pair of chromosomes having the same gene sequences, each one derived from one parent, paternal or maternal, father or mother, gametes. Okay, so when we apply this term homologous chromosome on this figure, we concluded that those are two homologous chromosomes, and these are also the same meaning or the same term, two homologous chromosomes. However, how can you differentiate between the first form and the second form of two homologous chromosomes? The shape of chromosomes which lies on your left hand, this is when you must express this is from father, gamete, and this chromosome derived from mother, chromosome, gamete. Okay, so both having the same genes. Okay, and when these chromosomes replicate by DNA replication at S phase. Okay, this led to H1 chromatid. This is called in general chromatid, and this is also called chromatid. Okay, chromatid. Please don't forget that. Each one of these chromosomes or chromatid duplicate their genetic material at the S phase during DNA replication. Each one will be forms another identical chromatid to each. Identical. So this identical for this, and this is identical for this. Okay, but the same chromosomes or homologous, having the same lengths and distribution of genes. This is maybe here, so maybe here, responsible for hair color, and this is also responsible for hair color, but this is derived from mother or father, as we mentioned, or father, and this is derived from mother. Okay, but both having the same genes, distribution of the same genes. Okay, so this is two sister chromatids, and those are having the same two sister chromatids. Okay, for example, to more clarifying this explanation, alleles different forms of the same gene. Okay, for example, this is an allele for blue eye color, and this is allele for brown color, eye color, and both are homologous chromosomes. This is derived from mother gamete, and this is derived from father gamete. Okay. Okay, it is worth to mention that the loci of this genetic trait represents the eye color also is found and or located at the same locus. Okay, so this is have the same locus for this is, but this is a B small and this is a for B capital. So this is a dominant character, and this is for recessive character. Okay. In turn, both chromosomes are called two homologous chromosomes. Non-homologous chromosomes are the chromosomes that are or don't belong to the same pair, meaning that both chromosomes have a different genes, not the same genes, have a different genes. Okay, we will explain this term in another slides or two alleles determine how hereditary trait is expressed in the cell. please be focused with me. This is two homologous chromosomes, okay, and each one is carried three genetic traits. For any hereditary characters. So the two chromosomes pair the alleles for each genetic or each characteristic. Okay, and those are called gene loci. This is gene locus, whilst all of them is called gene loci. Okay, when a letter is a capital, meaning dominant. When a letter is small, this meaning a recessive allele. Okay, let's start. When the genotype meaning or involving the two alleles of each gene, which may be both are capital letters, so these two homologous chromosomes are carried homozygous allele for a dominant gene characteristic. In another case, when both two alleles are similar also like that, but in recessive form, at which the two homologous chromosomes called two homologous chromosomes carrying homozygous recessive allele. Okay, the third case, when one of the two genes is in a capital form, so-called dominant allele, and another is in a recessive form or recessive allele, maybe PB, at which the two homologous chromosomes are called two homologous chromosomes carrying heterozygous dominant and one recessive allele. Okay, please, there is important noticeable. When in another fourth case like that, if a P capital here are found here, but the B or maybe B capital or B small are disappeared as a result of deletion for this chromosome at this part of this chromosome, deletion. So we called for this case, please think with me. Yeah, of course, this is a hemizygous, two homologous chromosomes carrying hemizygous allele. Okay, this is a fourth form of distribution of genotype like homozygous in a dominant case, or homozygous in recessive case allele, or heterozygous in one dominant and another in recessive case allele, or one is found and the other is not found, so it is called hemizygous. In more clarifying to the previous slide, we use such slide in front of you. Pairs of homologous metacentric chromosomes. Okay, like the previous. This is homologous to that chromosome. Okay, this chromosome are similar to this chromosome. That's right. So they called two homologous chromosomes. When we compare between these homologous chromosomes with another is also homologous to other. Okay, this is homologous for that chromosome. But when compared between these chromosomes and that chromosome, we concluded that both are a non-homologous chromosomes. That is and that's okay, because their shapes and their genes are different too. Okay, when say this is a gene for eye color, in turn this is also gene of eye color, which may be the same here and here, but is not necessary to having the same dominant or recessive. For example, maybe that is a T capital and that is a T small, whereas both having the same hereditary trait, but not necessary to have the same genes in allele. Have the same gene but not allele, haven't the same allele, like that, maybe like that or that. Okay, however, but when we compare between this chromosome taking green color with the red color, as previously mentioned as a non-homologous chromosomes, we concluded that this is a gene of length, but this gene is not here, maybe a gene of any character, eye color, eye color, it must be also in the homologous here, also eye color, but the length here and not here, and the eye is here but it is not here. Okay, so both green colors are called, which may be dark or light green, are homologous chromosomes, whilst the two red colors, which may or taking dark or light red color, are also two homologous. But the red color and the green color are not or non-homologous chromosomes. Okay? As well as both two arms are called two sister chromatids. Also, two sister chromatids. Two sister chromatids. And in turn, two sister chromatids. But when we compare between this chromatid and that chromatid, both are non-sister chromatids. In the same line, both are non the same two sister chromatids. Okay? Now we will study another point. Eukaryotic organization from DNA to a duplicated metaphase chromosome. This figure showing the condensation of DNA double helix like that. When this DNA double helix molecules wrapped around histones, okay, wrapped around histones. So this will be for nucleosomes coiled into a chromatin fiber. This is a chromatin fiber. Consists of nucleosomes. The DNA bind between them is called DNA linker. Okay. This form is called chromatin fibers. More condensation and further condensation of chromatin. This will be form this shape. This condensation accompanied with folding of DNA around histones proteins, and finally we form that shape of chromosomes. One chromatid. Such chromatid or chromosomes duplicated by S-phase, forming the another chromatid. So this is a duplicated chromosome or duplicated metaphase chromosome. This is a shape which can you detect it in the lab when you prepare the slides of chromosome for karyotyping or to study the chromosomal aberrations. Don't forget it. Chromosome, thread-like structure of nucleic acid, carrying genetic information as genes. Chromatid, two thread-like strands. Each contains double helix of DNA. Chromatin, consisting of protein, RNA, and DNA. Now we will study the most important technique called karyotyping. Such procedure is used to prepare a karyotype of chromosomes from any organism from the metaphase spread of chromosomes. So a karyotype is assembly or picture of an organism's chromosomes. The term refers to a laboratory technique that produces an image of an individual's chromosomes. In order to get this picture, the chromosomes are isolated from blood vessels in the case of human, white blood cells from human, and from bone marrow from the mouse or experimental animals. Okay, this is meaning isolation from, as we see, bone marrow in the case of experimental animals, and from white species from human. Then these chromosomes stained and examined under the microscope, then arrange each two homologous chromosomes next to each other beside them, and each group in rows according to their lengths. Okay, this depends on the position of centromere to compare or to collect each two homologous chromosomes next to each other. So the examining of these chromosomes through karyotyping allows the investigator to determine whether there are any abnormalities, maybe structural or numerical abnormalities or aberrations within this karyotype of chromosomes. Now we will show the some examples to illustrate this procedure. First example, this is the karyotype of mouse chromosomes collected from stem cell lines with a normal male karyotype. Male, so it contains the total number of chromosomes are 40 telocentric chromosomes. Don't forget, telocentric, all chromosomes of mouse are telocentric chromosomes involving 38 autosomes and XY sex chromosomes. Okay, you see this is a 2 homologous chromosome 1, 2 homologous of chromosome 2, and so on, 3, and so on. This chromosome is from male gamete, and this is from female gamete, and the other from female gamete. Okay, one from male gamete and the other from the female gamete. 20 from male gamete and 20 chromosomes from the female gametes in the case of mouse. Mus musculus, genus or species, Mus musculus. So we arrange these chromosomes into, as you see, into homologous chromosomes, each two homologous chromosomes next to each other. And finally, we ended with X chromosome and Y chromosomes, as you see. This technique is called G-banding. G-banding. G-banding, depending on the staining of chromosomes with Giemsa stain. Giemsa stain. This stain or dye is so famous to stain the chromosomes. Also, there is another staining called orcine, especially for plant cells. Okay, this technique depends on the appearance of chromosomes as a banding layers like that. Each banding in homologous chromosomes is similar to another chromosomes like that, like that. Okay. When the band, as you see, dark or light stain, this meaning the dark represents the heterochromatin and light staining represent the euchromatin. Okay, so this is a centromeric region taking as a permanent region, taking a dark staining to represent its structure of heterochromatin of centromeric regions. Type. Okay, please to go to another example. Example 2. Karyotype in male mouse stem cell line stained with G banding also, and revealing, as you see, also the another. In this case, we noted that the chromosomes, especially chromosome 8, taking 3 copies. This is a abnormal number or numerical aberrations called trisomy. Tri meaning 3. Rosomy repeat of these chromosomes in 3 copies, as you see. This is aberration or abnormality for 8 chromosomes. So the karyotype, as mentioned before, showing and determine the structural or numerical aberration. At this case, this picture showing the used to determine the numerical or number aberrations, as you see. Okay. In the third example 3, this is a technique, a modern technique, as following. Okay, this modern technique using a modified fish technique. This is more recently than the other G-banding classical. Chromosomal aberrations. Chromosomal aberrations meaning variation in chromosomal structure or number, forming that term, chromosomal aberration. They can refer to changes in the number of sets of chromosomes, at which is named ploidy. Ploidy form the most famous of chromosomal aberrations is polyploidy. Polyploidy. Okay. And changes in the number of individual chromosomes at which called somy, like that trisomy. In the case of Down syndrome, or changes in the appearance of individual chromosomes through mutation-induced rearrangement, rearrangement of some chromosomes or some parts on the chromosome. They all can be also associated with the genetic diseases or with species differences. However, there are many techniques are used to detect and stain the chromosomes to detect such chromosomal aberration, like that by FISH. FISH is abbreviation for fluorescence in situ, hybridization, and banding. Banding may be C banding or G banding. C banding meaning staining the centromeric region, and G banding meaning staining both heterochromatin and euchromatin on along the chromosomes. To use these chromosomes for karyotyping and detection of chromosomes. As described before, the chromosomal aberrations classified into two main categories: structural abnormalities and numerical abnormalities or aberrations. We start with the first category, which includes the following as you see in this split. The structural aberrations of chromosomes includes the following. We find some aberrations like that deletion meaning segment of one arm of the chromosome will be deleted, like this segment. Okay, so this segment in turn containing a specific gene sequence. So this gene is lost and the cell haven't the effect of this gene on the cell. The number one of the chromosomal aberrations. There are also ring chromosome, and so famous in metaphase spreads in most slides when you study the chromosomal structural aberrations. This ring chromosome, where the chromosome two ends fuse together by where both ends, which named telomeres, are deleted, then the terminal ends of this chromosome became sticky and in turn fused or spliced together. So this led to this aberration, ring chromosome. This is a centromeric region, and both ends fused together at here. Okay. There are also isochromosome. Isochromosome is a structural aberration, whereas there is a split at this region of centromere, leading to formation each two arms, one for up and the other for down, causing which we say isochromosome. Also, there is a inversion, whereas the some segments are up and the other are down, this is causing disturbance of the gene sequence. Besides, this is a derivative chromosome, whereas there is also a connection between the original chromosome and other part. Translocation, this is the structural operation which include that both chromosomes exchange their segments like that. This part exchanged with the this part of another chromosome. Also, this is led to the disturbance of gene sequence. Moreover, there is also insertion meaning part of chromosome of another chromosome fused with this original chromosome, which is taking green color, and the red color is a foreign segment of another chromosome which added to this chromosome, so may be called addition. This plate exhibiting 6 photomicrographs, all isolated from bone marrow of experimental mice, whereas the A and B and C, G, E, all containing the chromosomal structure, chromosomal aberrations, as we detected in these photomicrographs. We are say which these aberrations which included in this photomicrograph in respectively. Besides that, there is a photomicrograph in the middle containing chromosomes collected from normal mouse, taking normal appearance, so we use this photomicrograph for comparison to detect and investigate the other photomicrographs. Firstly, the photomicrograph A, we noted that there is deletion, whereas this chromosome have one arm is longer than the other, where the other is deleted segment from one arm, and the other so appeared more longer than the other. Deletion. Okay, this is a first structural aberration. In slide B, or this photomicrograph, we notice that the fragment which found, I will be marker this fragment, which may be deficient from one of these chromosomes. As described before, the total number of all chromosomes of mice are telocentric and equals 40 number of chromosomes. Okay, this is a fragment F fragment. In photomicrograph number C, we noticed that there is also a centromeric attenuation. Centromeric attenuation meaning that the centromere suffering from weakness, and so both two arms are separated from each other at the centromeric region, like this chromosome and like this chromosome. Centromeric attenuation. Okay, we detected this aberration, structural aberration in both chromosomes. In this photomicrograph E, we notice that there is a ring R, ring. So this chromosome, as we described before, the previous slide, these both ends of chromosome fuse together and forming a ring-shaped chromosome. Finally, in this photomicrograph number G, we notice that break B, whereas there is a break at this chromatid, so maybe called chromatid break at this chromosome. We noticed that break and indicated with this arrow. So this is the most structural aberrations we can summarize in 6. This plate represents the second category of chromosomal aberrations as numerical chromosomal aberrations. The most famous of such abnormalities is a polyploidy. Poly meaning multiple of polyploidy. Polyploidy such as when we say n chromosomes or n chromosomes at the case of mice equals 20. So this is called haploid. Okay, when this number equals 40, it is called diploid number of chromosomes. Diploid. Okay, haploid when you say 20 chromosomes in the field, and diploid when you detect 40 chromosomes in the field. This is this number represent the gamete of mice, sperm or ovum, whilst this is the normal number of cells. Okay, normal number of chromosomes in the somatic cells. This figure represents the normal appearance of chromosomes. So it must be contain 40 chromosomes. Okay, and used to compare and to identify the numerical chromosomal aberrations in the second figure. When you count the number of these chromosomes, you found that number equals 80 chromosomes. So these 80 chromosomes meaning that this is when 20 is a haploid, 40 is a diploid, and 60 triploid, 60 chromosomes, triploid. When it becomes 80 chromosomes like this slide, so this is a tetraploidy, meaning that n powered with a 4, okay, where n equals in the case of mouse with 20 chromosomes. So when it is powered with 4, so it is became the total number of chromosomes 80 chromosomes. Such phenomenon is called tetraploid, as one of the polyploid as a category of the numerical chromosomal aberrations detected in this figure. The mitotic cell division in animal and plant cells. This plate showing the recap of mitosis, which are included the following phases. Firstly, this prophase stage or prophase as first stage of mitotic division, at which the chromosomes condensed. So we are chromosomes are condensed. Prophase. P. M, abbreviation of metaphase, at which the chromosomes are found in the middle line of the cell or equator of the cell. So the chromosomes are found in the middle line or equator of the cell. The third one stage is called anaphase, A, meaning apart, whereas the chromosomes move apart from each other. So this led to a separation between the two sets of chromosomes. The last one, T, telophase, meaning two nuclei are formed at the end of this stage. These cells, which formed by the mitotic division, are identical to each other, having the same genes and the same alleles. This plate showing the different stages of mitotic division in animal or human cells. The mitosis passes through the following stages as illustrated in the different figures as you show. When you start with the first stage, which is called interphase stage, or before mitosis, we note that the chromatin coiled together. Okay, in early prophase, this the chromatin condensed more to form chromosomes. Okay, this is chromosomes. Okay, at prophase, the chromosomes will be appeared in a prominent shape, as you see. The fourth one is called late prophase. Now the chromosomes, you can distinguish between them. So we will go to for another stage, which is called metaphase. We will start with a transition of metaphase, chromosomes ready to arrange at the equator of the cell. And this is a more prominent of metaphase stage where the chromosomes are arranged completely at the midline of the cell. So after that stage, this is a anaphase where the chromosome each set found at the pool of the cell, and then the centrosome will be withdraw each chromosomal set in each pool, as you detect in this plate slide. After that, telophase, meaning that the each chromosomal set separate at least on the level of nucleus. So finally, this represents the late telophase, whereas the stage of cytokinesis appeared to separate the two cells or two identical cells from each other at the end of the two figures showing different stages of mitosis in onion root tips. You can detect some stages of mitotic division like that for to detect the first interface, like that cell, and this is also in interface. In prophase, like this cell also divides at the stage of prophase. So this is also in the prophase stage, whilst this cell at the metaphase, and so prominent to represent such a stage of mitotic division. And those cells that's beside this and this are represented the different stages of anaphase, like that. This is a pro or early anaphase, and this also in early anaphase, while this cell and this and that's are in late stage of anaphase. Okay. However, both cells represent the telophase and pass through the cytokinesis. Okay. Thus, the most important stages of mitotic division in onion root tips. You can conclude, depending the basis we are selected those cells at different stages of mitosis. You can also detect such cell, determine which the stage it passes, and that's all beside the cells. To examine, you can conclude the stages which these cells pass.